Okay, good morning, students. Today we'll be discussing about the types of fermenters or bioreactors in which the various industrial fermentations are carried out, processes are carried out to form the industrially important things and manufacture different things like alcohols, different solvents, enzymes, antibodies, amino acids, and etc. And also the fermenters which are utilized in the wastewater treatment and other such different varieties of processes. So basically there are different types of fermenters, and all of them are you have some different properties and different structural differences which are utilized for different types of processes. So we'll be studying about the different configurations of the fermenters and the basic bioreactor design also. So first of all, let us start with the definition of the fermenter. A bioreactor or a fermenter may refer to any manufactured or engineered device or system that supports a biologically active environment. For example, temperature, pH, dissolved oxygen, etc. So basically, it is a vessel in which the different biochemical processes are carried out involving the formation of the desired product, and that reactions or the conversion of the substrate to the product can either be aerobic or anaerobic. And in this engineered device or a system, the environment is controlled in a very precise manner so that the manufacturing of the product is can be controlled. So it can also be regarded as a containment system which provides the right and optimal growth, right environment for the optimal growth and metabolic activity of the organism. So let us have a look at the general features of an ideal fermenter. The first and important thing to note here is that the material that we are using for the formation of the fermental vessel should be strong and it should be resistant to corrosion. So basically it should be strong and inert substance, and it should be free from toxic effect from microbial culture. In the fermenter, it should be easy to control the contaminating microbes, and there has to be an inoculation point for that for the aseptic transfer of the inoculum. So basically in the fermental vessel, there is an inoculation point, that is a point or an inlet where the inoculum or the microbial culture is added to the system. In case of aerobic processes where oxygen is required, there has to be an aerating devices, and also there should be a provision for the stirring devices, that is for the uniform mixing of air, nutrients as well as the microbes. So there are things which are known as baffles, which are the rectangular plates which is attached to the walls of the fermenter, and they are they avoid the formation of the vortex. There should also be a sampling valve for conducting the laboratory test. From time to time, as the fermentation process or the biochemical production process passes on and goes on, there arises a need to check the sample as to what is the conditions inside the concentrations and different conditions and parameters inside the fermenter vessels. So sampling valves are utilized to take out the sample at different times and test them in the laboratories. There has to be a control of pH and temperature system in the fermenter, and also there should be a provision for intermittent addition of antifoam agent. There should be a drain at the bottom for removal of completed fermentation broth, and there should be a manhole at the top for repairing, thorough cleaning and other such activities. All the bioreactors actually deal with heterogeneous system, that is there are two or more phases. There has to be a liquid phase or gas or solid phase. Basically, there is more than one phase, that is either liquid gas or liquid solid or solid gas. So basically, these contains the heterogeneous systems, and there should be optimal conditions for fermentations, and there has to be efficient transfer of mass, heat and momentum from one phase to another. Generally what is done is there 20 to 25% of the volume is left unfilled at the top of the fermenter. That empty space is known as the head space. And what is the function of that head space? What is the uh, thing why we are leaving that empty space? Head space is to allow for splashing, foaming and aeration. So the fermenter designs varies greatly depending on the type and fermentation for which we are using it. So again, these are the simple things that a fermenter or bioreactor should have: agitation, aeration systems for oxygen supply, regulation of temperature, pH, pressure, nutrient feeding, liquid levels, etc. There should be provisions for sterilization and withdrawals of cells and mediums for continuous fermenters. And the more modern fermenters are usually, integrated with computer systems so that we can monitor them efficiently, and we can also have the data when we need it. So this is the diagram of a basic fermenter vessel, wherein you can see the central shaft, which is driven by a motor, and to this central shaft are attached these impellers. So these rectangular blades are known as impellers, and the function of the impellers is for the steering purposes and uniformity inside the culture vessel. This is the viewing port, and this is the pH controller, acid base reservoir you can see. Then you have the exhaust, cooling jackets. Now these are the baffles which are attached to the wall of the fermenter, and it helps in the avoiding the vortex formation. So this empty space is basically the head space. Now you can see the sterile air sparger through which the air is inserted in the fermenter vessel. So as we talk about the types of fermenter, basically, the mode of operations, as we all know that the fermentations can be carried out in different modes, including batch fermentation, continuous fermentation, fed batch reactions, recyclable reactors. So basically the configurations that can be utilized in different fermentations, the basic types of fermenters are the airlift bioreactors, tower fermenters, which include the type bubble column reactors, plug flow reactors, in which the system is not a liquid system, it is basically semi-solid system which is flowing, and it is a unidirectional flow. Then we have a packed bed bioreactors, fluidized bed bioreactors, and the stirred tank bioreactors, or the continuous stirred tank bioreactors. So let us start with the first type, that is the airlift fermenters. What happens in this type of fermenter, that the body of the fermental vessel is divided into two zones. All right, so there is basically the in the airlift fermenter, the liquid culture volume of the vessel is divided into two interconnected zones with the help of a baffle, or a you can call it as a draft tube. And there are two sections, the internal sections and the outer region, or the this is the upcomer and this is known as the downcomer. So only one of the two zones is sparged with air. We are passing the air in only one zone, that is the internal zone, and this zone is known as the sparged zone. So the other zone does not receive any air, and it is known as the downcomer. So this is the riser, the internal portion where we are sparging the air or the gas, it is known as the riser, and the outer portion where we are not inserting any gas or air is the downcomer. So the bulk density of the gas liquid dispersion in the gas sparged riser is a little bit lower than the bulk density in the downcomer. So therefore what happens is there is a flow, the dispersion flows up in the riser zone and it comes down through the downcomer. So basically the movement of the nutrient media is, it is rising up in the riser zone with the air and the gas which is being sparged, and it comes down through the downcomer. So it is basically getting uniformly mixed and with the help of the gas and the sparger. The airlift bioreactor are generally classified as pneumatic reactors because there is no mechanical steering uh, which is being done, and the turbulence is caused by the fluid flow ensures adequate mixing. Because of the fluid which is flowing in the riser and the downcomer, the turbulence is actually causing the mixing of the nutrients, microbes, and other substrates. The draft tube is provided in the central section of the reactor. The introduction of the fluid causes upward motion and results in the circulatory flow in the entire reactor. So basically there is a circulatory flow inside the reactor, that is from here it is rising up, again it will go down the downcomer, and again it will rise up in the riser. So there is a circulatory flow when inside the reactor vessel. The air liquid velocities will be low and hence the energy consumption is also low. The airlift bioreactors can be used for both free and immobilized cells, and there are very few reports on ALB for metabolic production. The advantages of this type of reactors are the elimination of attrition effects, and it is ideally suited for aerobic cultures since oxygen mass transfer coefficient are quite high in comparison to stir tank reactors. They are basically very high in energy efficiency, and they are often used at the large scales for the manufacture of biopharmaceutical proteins which are obtained from fragile animal cells. The heat and mass transfer capabilities of airlift reactors are as good as those of other systems. So basically you can see three different configurations of the airlift fermenters, in which we have the first is the this one where the draft tube is the internal loop configuration. The next is the split cylinder device where the cylinder is divided, where the one portion is be getting sparged and it is being separated, and this portion becomes the downcomer. And the third configuration is an external loop device where the central portion this act as the separator, and there is a loop which is formed inside the fermenter vessel, and there's a circulation of the media across this central region. So these are the three possible airlift fermenter configurations. The rate of this liquid circulation increases with the square root of the height of the airlift device, and as a result, because the liquid circulation is driven by the gas hold-up difference between riser and the downcomer, the circulation is enhanced or it is improved if there is little or no gas in the downcomer. So basically the focus is that there should be no hold-up of the gas or air in the downcomer. All the gas in the downcomer comes from being entrained in with the liquid as it flows into the downcomer from riser near the top of the reactor. So basically we are not asparging any gas in the downcomer. Any gas which is coming to the downcomer is because with the liquid entrapped, which is entrapped in the liquid coming from the riser zone. The next type is the continuous stir tank fermenter. Basically, these fermenters, the contents of these vessels are at steady state. That is, the contents and the concentrations of the different components of media and also the microorganisms, they do not vary with the time. They can be run in the chemostatic mode or the turbidostatic mode. Basically, these continuous system are employing yeast and bacteria to get the desired products as well as the primary metabolites, including enzymes, amino acids, and alcohols. These type of reactors are also, utilized for the activated sludge process for wastewater treatment as well as the production of the baker's yeast. There's a concept of washout which results if we increase the flow rate to a very high value. So basically, to deal with that washout conditions, there is an inoculation of the fresh microbial cells along with the substrate time to time. So this is a simple, diagram of a CSTR, continuous stir tank bioreactor, which is composed of a cylindrical vessel, and we have a motor-driven central shaft that supports the agitators or impellers. The shaft is fitted at the bottom of the bioreactor. The number of impellers can vary depending on the size of the bioreactor, that is the height to diameter ratio. And several types of impellers can be there, basically rustrum, disc, concave, bladed, marine propeller. So basically there are many types of impellers that can be present, and the air is added to this culture media with the help of the spargers that we have shown earlier. And these spargers may contain many holes or a single tube with a single orifice. The sparger along with impellers or agitators enables the better gas distribution system throughout the vessel, and also the bubbles which are generated with this sparger are broken down by the impellers into smaller ones, and it is distributed uniformly within the culture reactor vessel with the help of the impellers. The next type is the tower fermenter. As you can see, it is a very long fermenter which has a aspect ratio of height to diameter of 6:1, through which there is a unidirectional flow of gas. So basically, the gas is moving just in one direction, and see, we are inserting the oxygen from the bottom of the fermenter. It rises up in this direction towards the top of the fermenter, and it is getting converted into CO2 as the fermentation processes, and the carbon dioxide is released from the top of the fermenter. This is used for the continuous production of alcoholic beverages. One of the common type of, a tower fermenter includes bubble column fermenter, in which the air and the oxygen transfer, mixing, and other performance factors are influenced mainly by the gas flow rate and the properties of the fluid. Gas in this type of fermenter is sparged at the base of the column through perforated pipes, plates, or glass or metal porous spargers. The aspect ratio of height to diameter is 4 to 4 is to 6 in the bubble column fermenters. In the next type of reactors is the third type is the plug flow reactors. In this, it is very, the concentration of the substrate, or you can say the contents of the reactor vessel, is not same at all the regions. As the inlet or the substrate moves from one portion to the inlet to the outlet, the physiochemical, the chemical, and the physical properties of the plug, you can say, or the reactants changes from one region to another, and it is not uniform. So it is basically used for the treatment of the sludge which we get after the wastewater treatment, and it moves as the plug from inlet to from here in this U type of configuration from this portion to this portion. And plug flow reactors can be modeled as a pipe where the reactants move as a plug along the pipe. The concentration of the reaction, the most important thing is, in a CSTR, the concentration of the reactants is uniform across the fermenter, whereas in the plug flow reactor, the concentration varies. along the pipe, and there is no mixing between the beginning and the end of the system. There is no mixing between the different regions of the reactors. As these reactants move from the inlet to the outlet, they are treated and they are converted into the less toxic form. So basically, this type of fermenters are reactors are used for the treatment of sludge. The next type of bioreactors are the packed bed bioreactors, in which the solid media is utilized to form the immobilized bed, and within this within this solid media or the solid bed, the microorganisms are attached in the form of biofilms, or they may be attached to the surface of this packed bed, or they may also be present inside the particles and entrapped within these particles of the reactor bed. There may be some disadvantages of the packed bed reactors, which include the alterations in the bed porosity, bed compaction, and channeling. So basically, you can these column and the reactor beds can be packed with the enzymes, and the substrate can be added from the top of the reactor. And as the substrate moves with the from the reactor bed, in which the enzymes are immobilized, as the substrate move down the reactor bed, they are converted into the products. The next type of reactors are the fluidized bed bioreactor, which are developed for the biological systems that include various phases like solid, liquid, and gas. The fluidized bed bioreactor are generally operated in co-current upflow with liquid in continuous, and they can be of three types, in which the first is the inert core, on which the biomass is created by cell attachment. Second is the porous particles, in which the biocatalyst is entrapped. And the third category can be cell aggregates or flocks. So basically, in comparison to conventional mechanically stored reactors, fluidized bed bioreactors, they provide a much lower attrition of solid particles. And the advantage is that it does not have the drawback of clogging, channeling, and bed compaction. So that's all for today. Thank you.